So our next speaker is Professor Sitabhra Sinha. I mean, I think most of us know him. It would be like introducing Rajnikant to Tamil audience now, right? So in today's talk, he'll enlighten us on knowing when you're at the right size. It is about how do you apply global knowledge from local information to cellular assemblies.
Thank you. That was too much, actually, what you just said about me. Thank you. First of all, just like to express my appreciation for Arijit and his team of extremely enthusiastic students. I mean, hats off. I mean, he just organized this Network Biology Day just a month back, and now he's organizing this. I wish I could be as energetic. Arijit, great work. And whatever, you coordinated, which is actually in line with what I'm going to talk about. All right. So there will be a kind of a change of gear from the preceding two talks. It'll be more of talking about basic science, questions about development. There would be a nod to cancer, and there will be kind of connect to what Anu was talking about. But we are not really trying to figure out a practical way to solve it. It's pretty much so. But there would be kind of a very interesting connection that we would draw between development and cancer. Okay, and so more about it as we go through the talk. Oh, so very important. This talk essentially stems from the work of my graduate student Chandrasekhar, who's now doing his postdoc at Harvard, Shakti Menon, who's a senior scientist in my group. He was around yesterday, so some of you might have already met him, and Fernando Casares, who's a scientist at this Center for Biology of Andalusia in Spain. So as you might imagine, the talk title is actually a homage to this very famous essay by Holden written in 1926, in which he had very presciently actually spoke about how the size of an organism is often vitally related to the biology underlying it. Okay, so he said that ants are a very specific size because they use diffusion. You cannot just scale up an ant. I mean, forget about Hollywood movies where you have this giant, like ants, several hundred meters, which are devouring cities. That's simply not practically possible because of the biological impossibility of diffusion, which the ants use to essentially do metabolism. And so long before they attack their first city, the ants would just asphyxiate. By contrast, you can't scale down a human to the scale of an ant because, again, it will simply not be able to survive given the biology by which it kind of pumps blood and so on. Okay? But the reason why I'm so inspired by this article or the essay is that essentially what he pointed out is that every system, be it an organism, be it an organ, or be it some kind of a tissue, has an optimal size, which is governed by the underlying biology. So it must grow to precisely that size or a range of size and no more. And if you have a breakdown in this regulation of the size, then you're in trouble. In particular, for example, uncontrolled cell division would lead to cancer. All right? But also in the case of development, you don't want to end up with an organism which has a giant-sized head with a puny-sized body, right? So we rarely see such abnormalities from happening. So the question we ask is, how does the cell know when the tissue that it is part of has reached the right size? And connected with this question is that given whatever information it gathers from its immediate environment, how do all the cells of this tissue switch off growing further by division in a coordinated fashion? So it's no good if certain cells continue dividing while other cells have stopped dividing. So they also need to coordinate. Right? So the fundamental question we're asking is, A cell has only local information about its neighborhood. So the most it can hope to achieve is by looking at the cells all around it. But then through this, it has to acquire a global information about how large the assembly of which it is part of is, whether it be tissue or organ or whatever, and accordingly take appropriate decision. So if it figures out from just local information that the tissue has grown to this particular size, it has to decide whether to switch off further division or not. Okay? And just to give you an analogy, this is like you being part of an enormous crowd and trying to figure out how large is the crowd. Okay? So this is of course from the very popular children's, actually not just children, a lot of adults also enjoy going through this, called Where's Waldo? So Waldo is this particular character, and you have to basically search where is Waldo in all of this, but it also illustrates this idea that if you are somewhere over here, how can you actually figure out precisely how large this crowd is, because you can only see immediately around you. Okay? And another analogy comes to mind. So when I was a graduate student, I had lots of time to waste. So for a while, I used to play tabletop war games with a friend of mine. So we would recreate Waterloo, and I'd be, let's say, commanding the English side, my friend would be commanding the French side, and then we'll take turns and so on. And inevitably, most of these games, the French side won. Okay, but you know the real history of Waterloo, right? Napoleon lost. So doing this over and over again, my friend and I were very puzzled. Why is it that in our tabletop war games, the French were always winning? Well, one reason is, of course, that the French outnumbered the British at the beginning of the battle, right? So the Prussians, led by Blucher, who came in the evening, eventually turned the tide. But Napoleon had lots of opportunities to actually, beat off the British, before Blucher came anywhere in the horizon. So we were very puzzled, like for example, one of our favorite tactics would be to, so we are commanders, right? So we can right now see like where each division is. So there is no fog of battle, right? So essentially what we would do is we would send our best, division, let's say the Imperial Guards, to capture Wellington's command post. So once Wellington is out of the picture, the British army basically dissolves into chaos. And so we were always like figuring out, Napoleon was a great commander, right? But why didn't he win Waterloo if we, like bozos who have no clue about directing real battles, are always winning this? So then I came across this brilliant book by John Keegan called The Face of Battle. And The Face of Battle is basically looking at war, but not from the perspective of the big commanders, but from the perspective of the foot soldier or the cavalry soldier. So he looks at exactly the same region in space, namely the French-Belgian border, and he looks at three battles: Agincourt, done in the medieval age, where the British and French fought and the British won; Waterloo, where again the united forces of Britain and Germany fought with France, and again the Allied forces won over France; and finally, First World War. I think it was Somme or something, which was fought there. So what he's talking about is that, of course, the technology that you're using in each of these battles, one has been fought in Middle Ages, one has been fought in the early 19th century, and one has been fought in the early 20th century. The technology changes. The command and coordination ability to coordinate huge forces have changed. So actually the war is now taking place over larger and larger ranges. So Agincourt was just basically a football field. Waterloo was taking place over a few scale of villages, and the First World War battles of Somme were basically taking place over almost a province, right? So that's because of the advances in technology. But from the point of view of the foot soldier, it's the same chaos. A foot soldier has no clue what's going on in the battle. Typically it's a he. I know it's very politically correct now to say he or she, but unfortunately in those battles it was always a he. So you basically are only able to look at, let's say, the people around you, and of course your enemy right in front of you. And that's about all the information you get, okay? And it's a complete chaos, okay? And the only thing that you are thinking about is survive, right? It's only the big commanders like, Napoleon on the hilltop who's like, looking through his telescope and trying to, make some, sense out of this mess and trying to control this a bit, but it's not really controllable, okay? And in that analogy, essentially, this problem becomes the cells are in the foot role of these foot soldiers. They're trying to make sense, but there's no Napoleon out there. So they have to do all of this coordination themselves, right? So how do you do this without there being a overall commander coordinating all of this? So that's essentially the scientific question, okay? And interestingly, it turns out that there is another scale in which the same problem occurs, and that's in quorum sensing. So in quorum sensing, what you have is that you have bacteria which are, essentially, let's say, gathering forces, so to say, in some part of a host organism. Now, they can't show their hand too soon, okay? So initially they would like to, hide their true intentions and infiltrate, because if they show their hands too soon, the host immune system is going to just clobber them. But eventually, they would like to reveal their true faces. So the question is, at what point do you do this? Well, I think all of you know that it's when they realize that they have gathered enough forces that they can overwhelm the forces of the host's immune system. Again, the question is, how do you know? A bacteria is like, an individual cell in a small corner of the whole field, so to say. How do you know that your forces are outnumbering that of your enemy, namely the host's immune system? Okay? And so that's the whole problem of quorum sensing. The individual bacterial cell has to figure out that its brethren are enough in number so that now if we switch on our hostile intentions, there's nothing much that the host immune system can do. We can overwhelm it. Okay? And there have been several mechanisms which have been proposed as to how the bacteria can do this. For example, one of the mechanisms that is proposed is that maybe what they do is that they just release chemicals which act as signaling molecules into this extracellular medium, and the other bacteria kind of sense it, and then they in turn release some chemicals. And so if each bacteria is acting as a receiver and a sender, so if you have enough chemicals accumulated in the extracellular medium, just by sensing how much of this concentration is out there, you can get a sense of what's the size of the crowd. Okay? But that need not necessarily translate into other scenarios, in particular, the problem that we are interested in, which is how to arrest growth when the tissue or the organ has reached the appropriate size. So what's the question over here? So how do individual organisms or for individual organs, or for that matter the entire organism, know that they have attained their appropriate size and hence stopped growing? And of course we all know that if you fail to grow, then in the developmental scale you have deformities which could be potentially fatal. So this is a schematic picture of an overgrown Drosophila imaginal disc. And this is actually how it looks in the normal circumstances. So you can see that it's grown to some pretty weird kind of abnormally large size. And another side of this coin is that if you look at it once the organism has fully developed, so in its adult stage, then also unchecked growth can lead to problems. In particular, it can lead to cancer. So cancer and developmental deformities are, from this perspective, just two sides of the same coin. So how do we control growth? How do we make sure that it does not overgrow? So we have to somehow stop the cell cycle which regulates mitosis and therefore leads to cell division. So we know that we all start our life with a single cell. Then the cell divides into two. then each of those 2 cells divide into 4, the 4 divides into 8, 8 to 16, and so on, right? So if left unchecked, of course this will go to infinity, but it doesn't go to infinity because of lots of reasons. One of them being, of course, you've got finite resources. The cells need food, right? So long before you go to infinite, you'll run out of food. But even prior to that, there would be some appropriate size, and you need to figure out some way where it knows that it has to stop growing, even though it might have adequate nutrition. Okay? So in order to prevent, further cell division once it has reached the appropriate size, you need some way to arrest these oscillations within the cells, which is essentially the hallmark of cell cycle, in a coordinated fashion. And we know that this is essentially governed, these oscillations are essentially governed by cyclin proteins, which govern the transition between different stages of the cell cycle. So, I guess most of you are very familiar with this, but just so that we are on the same page, the cell cycle is essentially this periodic manner in which the cell undergoes mitosis. So, as a result of mitosis, let's say a cell is born. So then it undergoes a series of phases, which includes this G1 phase, then a DNA replication phase, then another phase of cell growth, G2, and then finally it divides into 2 daughter cells. And so what is happening is that if you look at the cyclin genes, they're undergoing oscillations, so that after completing one full period, you'll have one complete cycle completed, and the original daughter cell now itself becomes a mother cell and divides into 2. Okay? And so what we need to do is somehow stop this clock. Okay? So the question is, how do we stop that clock? But it's not just a question of just stopping the clock. Maybe you can stop the clock individually at a particular cell. How do you stop the clock at the right time? That is, when the cellular assembly has reached the right size. Okay? So this is the question we are trying to address. So for this, obviously we need somehow the cells have to exchange information. The cells have to communicate. But we have already said that the cells can only communicate with their immediate neighborhood. So what are the various mechanisms by which cells gather information about their neighborhood, or cells kind of communicate with their neighboring cells? So broadly, you can think about 3 mechanisms. One is diffusion. Okay? The problem with diffusion in our context is that it's pretty rare for large biomolecules like proteins and mRNA to diffuse between cells because of the plasma membrane which separates the cells. So each cell is kind of bound by this boundary of this plasma membrane, typically a bilayer lipid membrane. And this is kind of acting as a barrier for messenger molecules like proteins and mRNA to go through. So instead, the multicellular organisms have developed other techniques. So for example, like gap junctions. So gap junctions are specialized proteins. So there are various kinds of connexin proteins which combine together to form essentially tubes which connect the inside of one cell with the inside of a neighboring cell. So these are actually literally bridges which allow large molecules to go through from one cell to another. So this is a very common mechanism for short-range communication, for example, in the heart. Okay, so when you have myocytes in the heart, the muscle cells in the heart, which need to, of course, coordinate their contraction, the way they communicate is primarily through such gap junctions. But you can have yet another mechanism where instead of actually having something, some kind of a bridge directly connecting the cytoplasm of 2 cells with each other, you could instead have receptors on the membrane of a particular cell binding with the ligand of, the corresponding ligand of a neighboring cell. And when you have a suitable binding, This allows a series of downstream chemical reactions which causes certain activity to happen. Okay? So this is a receptor ligand mechanism is a very, very common strategy that many cells use to essentially have information about the neighborhood influence what is going to happen inside them. And one of the most common of these ligand receptor mechanisms is the so-called notch pathway. So the notch has been implicated in so many of the cell's functions that one biologist I heard once started his talk by saying there are two kinds of biologists: those who are working on the notch pathway and those who don't know they are working on the notch pathway. So let's try to look at how, by using this notch pathway for communicating between cells, we can actually propose a mechanism for arresting the growth. So the simplest scenario would be, let's say your cell density is initially pretty low. Okay? So in that case, most cells are not directly in contact with each other. As the cells divide, the cell density starts increasing, and as a result, the crowd, because of the crowding, the cells are much more likely to be in contact with each other. So finally, as more and more divisions happen, you go from a situation where the cells are pretty much not in contact with each other to where there is some intermediate level of contact. And finally, they are like Chennai public buses during office hours, right? You are like pushed against the cheek of your co-passenger, right? So you are being hemmed in from all sides, all right? So what we are doing is we are looking at the behavior of a cell cycle oscillator. So many of you may have heard of this repressilator, right? So it's a model which was proposed by Leibler and co-worker long back, but we now know that there are actually, in fact, many such repressilator-like motifs of three genes which are mutually inhibit each other in a cyclic fashion. So you have gene A, which is producing a protein which inhibits the expression of B gene, which in turn produces a protein which inhibits the expression of C gene, and C gene in turn inhibits the production of A gene. Okay? So the main difference with the model repressilator is that we have an auto-activation over here. So A can promote its own further activation. So this motif acts as an oscillator. So if you look at how the concentration of the A, B, and C gene products change over time, you'll find that essentially they show beautiful oscillation with A, B, and C's peaks shifted apart by 2 pi by 3. Okay? Now, this is the behavior of the individual genes, the genes in the individual cell. So, if you look at the equations, so, I don't want you to focus on the equations in detail. You would see that, this is primarily the rate of change of A is primarily governed by the concentration of A and the concentration of C, because C is the inhibitor of A. The B's rate of growth is governed by essentially A, because A is acting as the inhibitor. And finally, C's behavior is governed by B's repression. Now, you see that there's something in addition over here. And this is how essentially the cell talks to other cells. So what is happening is, here you have this receptors and ligands which are colored in red and blue. So when there's a receptor ligand binding, this produces a signal, okay, which we can call the contact induced signal. So this in turn will trigger a signaling cascade which eventually would have an inhibitory effect on the expression of the gene C. So I draw this with a dashed line because there could be some intermediates. It need not be that S immediately inhibits C, it could be through some intermediates. And that is precisely what is being modeled over here. So you can see S has a inhibitory effect on the production of C. So you see this parameter, which in hindsight we could have given a better name to it, but when we started off we didn't know that this was going to be the primary important thing. So this was, so we had various constants like K1, K2, K3, K4. So just by happenstance, this came to be known as K sub 8. Okay, but actually this is the one which is going to be the key parameter in our system, because it is basically telling you how strongly is one cell affecting the behavior of the other cell. So how strongly, so like if you think of it as shouting, so how loudly is one cell shouting to the other cell, okay? And that's going to be the key parameter. The other key parameter is, of course, the strength of this contact-induced signal, okay? And so what we want to see is that whether what we show schematically over here, that is, initially when you have low cell density, you have the cells cycle kind of oscillating in its usual fashion, and then with a little more increase in density, the cell cycle becomes slower, and then eventually at some critical density, the cell cycles completely stop, okay, and you've got a time invariant behavior. So it turns out that the simplicity of this model allows us to actually calculate this analytically. It's pretty rare to actually find a biological system where you can find a closed form analytical expression, but Chandrasekhar actually figured out that, look, if you can take this Hill functions, so Hill functions, if you have this Hill coefficients to be very high, they go from a sigmoid to essentially a step function. Okay, so step function means that if the value is less than this half saturation constant K, it will be completely switched off, and if it is greater than K, it will switch to its maximum capacity. So let's say it goes from 0 to 1, so it acts exactly like a step function. So once you actually go to that limit where you replace each of these Hill functions by a step function, things become very simple. Why? Because now you can just talk about the state of each of these cells as just a string of 3 numbers, which could be either 0 or 1. 0 means it's not expressing, 1 means it's expressing. So given that we have 3 genes, A, B, and C, the state of each cell can be determined by a string of 3 numbers. Is A 0 or 1? Is B 0 or 1? Is C 0 or 1? So I can just take a vector made of 3 components, which can go from 0, 0, 0 to 1, 1, 1. And so these 8 states. So why 8? Because each of these genes can be in 2 possible states, and there are 3 genes, so 2 times 2 times 2 makes 8. So there are just 8 possibilities, 8 possible states in which a cell can be, right? So now that we have reduced the cellular states to 3-bit binary strings, we can actually ask, okay, for various values of this strength of coupling of communication between the cells, what kind of dynamical transitions do we see in each of these cells? So it turns out that there is a critical value of this coupling strength, which you can actually analytically derive. So these are functions of the total concentration of the C gene, for example, the half saturation constant, and the signal. So if it is less than the critical value, then the cells kind of undergo this cyclic path. So it goes from 0, 1, 1 would mean that A is essentially switched off, B and C are switched on, then here only B is switched on, and then after a while it goes to a situation where A and B are switched on, C is switched off, then only A is switched on, and so on. So basically it undergoes a cycle involving this 6 states. The other 2 states basically form a 2 cycle, which is very hard to get at, so essentially its Basin of attraction is so negligible that you can actually forget about it. But once you switch your strength of coupling to the value beyond this critical value, you see that it immediately transitions to a completely different behavior, where these states essentially just converge to a globally attracting steady state, which involves only A being switched on and the other genes being switched off. So you have essentially stopped the cell cycle and the cells have stopped dividing. Okay? So you can see pretty easily that how just changing the strength of the intercellular coupling, you can go from a situation where you have cells undergoing regular oscillations and so therefore dividing, to a situation where the cells have stopped dividing and you have growth arrest. And this is pretty robust in the sense that, you might say, okay, you did all this, sleight of hand where you replace the Hill functions with step functions. How do I know that the real model still works like that? Well, if you replace the original cell cycle model's Hill function, the reduced model, you see that the pattern of oscillations is somewhat altered, but the range over which you get oscillation arrest in the space of parameters given by the contact-induced signal and the feedback strength or the communication strength between the cells, those pictures remain more or less intact. So this is the full cell cycle model. This is the reduced model in which we have used step functions instead of the Hill functions, and this is what we derive analytically. So the key parameters over here are the strength of communication between the cells and the intensity of the contact-induced signal, which you may recall increases as the cell density increases. So eventually, if you take any value of this strength of communication, you realize that after a critical signal, that is after you reach a critical density, the oscillations which regulate cell growth would be arrested and you will stop dividing. So the color represents the oscillation frequency, which is scaled so as to between 0 and 1. So 1 is the maximal frequency, 0 is like not oscillating. So you can see that it pretty much behaves pretty similarly even in the analytical expression compared to the cell cycle model. But now we kind of started asking a slightly different question. We said, okay, this is fine. It's also simple to understand that as density increases, you'll have more signals coming, and that can actually induce you to go to cell arrest or growth arrest. But what if you look at confluent epithelial sheets? So where's the problem? The problem is that unlike the previous system I told you about, in an epithelial sheet, like for example our skin, the cells are always in contact with the neighboring cells. So you can't argue that, they stopped dividing because now they have many more cells talking to them. Well, they're always in contact with other cells, no matter what stage they are in. So how is it that in the case of a confluent tissue where the cells are always in contact with other cells, the cells are basically forming, like a fully in touch kind of system, how do you actually have cellular arrest? So they must be using some other kind of mechanism. So, yeah, do I have 5 minutes or? Okay, couple of minutes, okay, good. So one of the things we realized is happening is that as the tissue grows, you go from a situation where the morphological characteristic of the cells, its size and shape, actually change so that you go from something which looks pretty disordered like this to something which is very ordered like this. So the reason this is happening is that here the number of cells is relatively low, so you have a lot of freedom in having various kinds of sizes and shapes. So the number of neighbors you have can also alter. So you can see that some cells are slightly larger, and so they have more neighbors. Some cells are smaller, so they have fewer neighbors, to a situation where the cell density is so high that you basically go to this tightly packed geometric structure. And that the most efficient way of packing cells is the hexagonal close packed lattice. So eventually it will go to a hexagonal kind of close packed structure. So what we find is that this leads to the reduction in the heterogeneity in the number of neighbors of a given cell with time as you go from the early stage to the late stage. So the question is, can this trigger the cell growth arrest? And we find that as you increase the homogeneity, so the more and more cells are added, you actually find that the distribution of the perimeter length of the cell becomes less and less broadly distributed. So this is the distribution of the cell perimeter length, and as you can see, it becomes tighter and tighter with the increase in the density of the cell. And so what this results in is that the strength of the contact-induced signal, which is itself a function of the perimeter, because we have assumed that the ligand concentration of this bound to the cell is proportional to its perimeter length. So this strength itself is now a function of the perimeter length. So what we find is that if we look at the growth rate as a function of the coupling strength of the contact-induced signal and see how it changes as we increase the homogeneity, so that means that as time progresses, more and more cells are added and the system becomes more homogeneous, you find that the tissue growth rate actually varies very systematically. But that's not the end of the story. It turns out there are two regimes. So if you have low coupling strength versus if you have high coupling strength, you could have completely different behavior. So because of lack of time, I will just skip across the details of it and just tell you that what is happening is that for strong coupling, as you increase the cell density, and so the system becomes more homogeneous, the growth rate actually decreases eventually to 0. And this is precisely what we are talking about, that there is cell division over here at initial phases, and then the cell division stops once you have enough number of cells and the system has become homogeneous. But there is another parameter regime which we call pathological, where at weak coupling the growth rate actually increases with increased homogeneity. So here, as you pack more and more cells, you actually have the growth rate increasing. So this in some sense is similar to what we see in tumors, for example. And so this is just my conclusion slide, that actually we found that we can generalize this to explain a lot of other things where contact-mediated signaling can enable disorder-driven transitions in cell assemblies. So where gradually go from disorder to order. So we can explain this to, for example, explain how you have this very regular arrangement of hair cells occurring in for example, the avian bacillar papillae, but also in various insects. So in a certain cells choose to become hair cells, other cells become support cells. And you can also see this, you can also use this to explain, for example, in the organism Trichoplax adherens, which has cilia. So the cilia kind of show this very regular beating. So one of the mysteries is that how does it propel itself? So to propel itself, some of the cilia must stop beating and others must continue to beat. And we find that by changing the disorder of this geometry of the cell-cell contacts, actually we can make certain groups of cells freeze and other groups of cells continue to beat because of the same oscillation arrest versus non-oscillation arrest. And so I'd like to thank the people who did this work. So as I mentioned, Chandrasekhar, who finished his PhD, I think just about 2 years back, and is now has a joint appointment at Harvard and Howard Hughes Medical Institute. Shakti Menon, who's a senior scientist in our group and is currently at IMSC, and Fernando, who's at this Andalusian Center for Biology at Seville. And of course, generous funding from Department of Atomic Energy has helped us all these years to carry out our work. So thanks.
Thanks a lot, Sitabhra. So we have time for only one question. The rest we can discuss during the coffee.
Yeah.
Hi.
So please go on.
Okay. Hi. So the question is that given your line of thought, how would you now think about the growth of a duct, say for example, where the duct is growing, but you are not allowed to change your, in a given cross section, you are not allowed to change the number of your neighbors. But at the same time, the duct does know that, okay, after I reach a particular limit, I need to stop growing. Like, how would these systems translate to those kinds of more constrained spatial structures for different organs and so on?
Okay, so when you say duct, you mean basically a one-dimensional geometry. So each cell has only two neighboring cells, right? That's what you mean. Yeah, very interesting question. I suspect that it would still be something to do with, the length distribution of these cells, but, we have only been looking at 2D geometries for obvious reasons, and it also translates to 3D, but the 1D geometry is something we have never explicitly looked at. But, I would be very interested to, discuss this with you maybe during lunch on this.
Yeah, thanks a lot, Sitabhra.